Now, spray drying is used by industry to produce foodstuffs, so things like milk powder or instant coffee. They do this because by changing the way in which a liquid solution is dried, you can control how the powder that is produced will behave. For example, shown here are particles made from the same glycoprotein solution. So, even though the particles are made of exactly the same composition, they will have different physical properties. For example, how fast they dissolve in water will be different. Now, the way they made these different particle structures from the same starting solution was by simply changing the temperature in which the aerosols were dried. By increasing the temperature, they increased something called the Peclet number. Now, the Peclet number simply describes the ratio of how fast the aerosol shrinks to how fast the insoluble particles in the aerosol diffuse. So if the Peclet number is high, the insoluble particles move slowly and thus are trapped on the surface of the aerosol as it dries. Particles will eventually form a shell on the surface of the aerosol, which then collapses as the remaining water evaporates. This results in the particles having these unusual shapes shown on the right. If the Peclet number is low, then particles will diffuse faster than the aerosol shrinks. This results in the particles not being enriched on the surface. This results in particles being formed that are more spherical, as shown here on the left. Now, what is a virus or bacteria if not an insoluble particle that is present in an evaporating aerosol of respiratory fluid? So shown here is a respiratory aerosol of PBS, which is essentially a saline solution that contains E. coli, or a bacteria, that was dried at 30% relative humidity. So the shadows on the surface of the particle are bacteria that are locked at the surface of the crystal. Now, based upon our understanding of spray drying, this is exactly what we would have expected. In dry air, bacteria, such as E. coli, would be expected to be accumulating on the surface of the evaporating water droplet. Now, collectively, what this means is that we are using our understanding of industrial food production to inform our studies into airborne microbe decay.